Hi, my name is Jaime Winternitz, and today I'll be talking about the genes of attraction. Mate choice for genetic benefits via mate preference or post-mating cryptic selection, while controversial, is believed to occur across diverse taxa, from vertebrates to invertebrates. But mate choice for genetic benefits is puzzling because as sexual selection for good genes reduces variation in the population, the incentive to be choosy also disappears. In essence, when all potential mates are of equal quality, why put costly effort into being choosy? Thus, mate choice should disappear since it no longer brings any benefits. An intriguing solution proposed by Hamilton and Zook is that genetic variation in condition-dependent traits preferred by females is maintained by continuously evolving parasites that change the relative superiority of resistance alleles. Besides the model of Hamilton and Zook, other models of mate choice for genetic benefits have also required fluctuating environments or external perturbations in gene frequencies, which can be equated with the effects of parasites, to maintain genetic variation at condition-determining genes. While many factors in a host environment change over time, few do more rapidly or consistently than genetic identity of a host's parasites. Parasites, broadly defined to include macroparasites like helminths, arthropods, and protozoa, and microbial pathogens like viruses, bacteria, and fungi, are ubiquitous in wild populations. Parasites can exert strong viability selection, exemplified by deadly epidemics, but more commonly are constantly present and exert selection on their host fecundity, which can be equally important to fitness. Parasites also fill the role of fluctuating environments, a requirement of most models of mate choice for genetic benefits. Immune genes are perhaps the best candidates for genetic quality models of mate choice for several reasons. First, they include some of the most polymorphic genes in the genome of vertebrates and invertebrates, suggesting their role in mate choice could be general across animals. For vertebrates, the major histocompatibility complex, or MHC, stands out for its unparalleled diversity, which can be seen in the genome. This figure shows the SNP density across human chromosomes, and the MHC region stands out on chromosome 6 for its polymorphism, and only regions of extensive structural variation can compare. Second, diversity at many immune genes is thought to be maintained by pathogen-mediated balancing selection, and similar forms of pathogen-mediated selection apply to those immune genes that are conserved across birds, mammals, and other vertebrates. Third, male condition often depends on infection status, providing an honest signal for heritable parasite resistance alleles that increase offspring viability and assumption of good genes models. My co-author Jesse and I conducted a systematic literature review to oversee the scope of studies on immune gene-mediated mate choice. We had three criteria for relevance. The study, 1, uses or discusses animals, in effect not plants, bacteria, or fungi. 2, characterizes immune genotypes, for example, sequencing or expression analysis of specific immune genes of study subjects. And 3, tests for mating behavior or sexual selection related to immune genotype. We were interested in empirical studies only, thus we excluded reviews, pure modeling studies, and meta-analyses that used previously published articles. Searches were conducted in Web of Science. For included studies, we extracted the following: the category of gene investigated, in essence, MHC or non-MHC, the number of genes investigated per category, species name, taxa, and study year. We followed PRISMA reporting guidelines, and this figure is a diagram showing the stages and records retained at each stage. In total, approximately 78% of studies, out of 92, investigated MHC genes, and only 22% looked for non-MHC immune genes. From the figure, we can see that in A, the MHC was the first to be investigated for its role in parasite-mediated sexual selection, and studies beyond this gene family were minimal over the following decade. From 2007 onward, the search for immunity genes beyond the MHC expanded, and the number of those genes is now nearly as rich as those targeted within the MHC. In B, we see that MHC studies typically focus on a small number of genes, whereas those beyond the MHC typically involve wider screening techniques, presumably due to the more exploratory nature of studies hunting for less well-described immunity genes as targets of sexual selection. In C, we see that the number of immune genes targeted beyond the MHC are comparatively enriched in mammals and birds and completely lacking in amphibians and reptiles. Focusing on the collection of studies that investigated non-MHC genes in relation to sexual selection and mate choice, some themes are apparent. Almost half of the 20 studies involved insects, mostly fruit flies, but also ants and beetles. These investigated between 5 and 14 immune genes, typically looking at differential expression related to male and/or female genotype. Many looked for signatures of positive selection on reproductive protein genes that have dual functions in immunity, speculating that female immune responses may assess sperm quality or compatibility. In contrast to lab-based studies on insects, those on birds and mammals were mostly field-based and looked for a wider array of immune genes and their associations with reproductive success, testosterone and parasitism, and patterns of non-random mating. A number of studies use multi-species comparisons to test evolutionary predictions. Those on primates and wild mice tested if the rate of immune gene evolution correlated with mating system under the expectation that higher promiscuity would increase exposure to parasites and thus select for faster immune evolution. While the most well-studied immunity genes for mate choice are by far those of the vertebrate MHC, the generality of the involvement of immunity genes extends to those underlying other functions of vertebrate immunity and to those acting in invertebrates. The omics age has facilitated investigation beyond candidate gene sequences, including de novo candidate gene approaches interrogating whole genomes, comparing differential transcription, and comparing differential protein abundance for putatively involved genes. Once candidate genes are identified, studies should take care to fully test assumptions of genetic benefit models by measuring mating preferences, measuring offspring viability, distinguishing between additive and non-additive benefits, and controlling from the manipulation of chooser investment. The greater the diversity of animals and populations in which these processes can be tested, the easier it will be to find prevailing patterns that can guide ongoing research. One practical justification for research on parasite-mediated sexual selection is that sexual selection is believed to increase population viability by promoting good genes and filtering out undesirable phenotypes from the population. This can occur, for example, from purging of deleterious recessive alleles in small inbred populations, like that of the Isle Royale wolf, which was originally founded with just 2 to 3 mainland individuals. A recent meta-analysis considered experimental evolution over multiple generations found that sexual selection does indeed tend to increase population fitness, particularly when populations are exposed to novel environmental conditions. However, they also found that increasing the strength of sexual selection typically resulted in comparatively weaker immunity in the population. These findings might be explained by a trade-off between sexually selected phenotypes and immunity, and they highlight that evidence for parasite-mediated sexual selection does not necessitate that populations will generally be more resistant to infectious disease. Going forward, there are deep implications for parasite-mediated sexual selection for conservation, wildlife management, and long-term viability of populations. It is important to consider that models of mate choice for genetic benefits and contextual factors, such as conflicting selection pressures and cases where genes influence multiple traits and/or interact with multiple genes, will complicate identifying immune genes involved in mate choice. With that, I'd like to acknowledge my funding sources, my collaborator Jesse, and thanks to the editors for the book Animal Behavior and Parasitism. And thank you for your attention.